Welcome to our webinar presentation today. I'm Peter Hoffelens, the publisher of ADC Review, Journal of Antibody-Drug Conjugates, and I'm your host today. In our presentation today, we will offer a comprehensive overview of findings showcasing how Slope Spectroscopy using the SoloVPE system has emerged as a dependable analytical methodology for supporting the quantification of protein concentration in antibody-drug conjugates, or ADCs. So what can you expect to hear today? You will gain insights into the challenges of traditional spectrophotometry when determining protein concentration in ADCs. You will discover an innovative variable path-length spectroscopy method designed to overcome these challenges. ADCs validation process will be shared, demonstrating the reliability of the SoloVPE system for precise protein concentration quantification in ADCs. Our speakers today are Joe Ferraiello and Priya Patel. Joe leads the Repligen bioanalytical application team, overseeing the variable path-length spectroscopy solution for inline and at-line applications. Drawing from over a decade of hands-on experience in analytical applications, Joe plays a pivotal role in enhancing the company's customer success. Joe's leadership ensures successful validation, smooth method development, and the integration of highly accurate process analytical tools into their biologic platforms, addressing critical production stages and bolstering their capabilities in biotechnology. Priya is a scientist at AbbVie, focusing on high throughput and concise method evaluation and development, using screening technologies for biologics, including monoclonal antibodies, ADCs, and multispecifics. Throughout her career, first as a quality control chemist, then as an analytical scientist, Priya has gained valuable experience in detailed method validation, understanding of GXP regulation, and developing novel methods for challenging biologics. Now, before we go, I'd like to ask the audience to please submit their questions for our Q&A session, which will be held after the presentations are completed. And with that, I'd like to invite Joe to come and share his first presentation with us. Joe?
Hello, everyone. Thank you, Peter, for the introduction. My name is Joe Ferraiello. I am the Director of Bioanalytics Applications for Repligen. I would like to welcome you all to the ADC Review webinar with Repligen and AbbVie today. My portion of the webinar will introduce the CTech SoloVPE technology and its benefits, how it works, and I will also introduce our approach for the ADC application using our system. We'll show you how you can implement dilution-free analytics to reduce deviations, especially when using the technology as a platform, how you can adopt Slope Spectroscopy across multiple sites and groups. At the end of the webinar, we'll have a live Q&A session where Priya and I can answer all of your questions. Repligen offers innovative solutions for all key stages of bioprocessing, from upstream intensification methods using ATF and TFDF through chromatography. Systems focus on improving high yield, as well as our well-known OPUS columns. Continue to provide systems that improve productivity in the downstream process filtration and formulation systems that are scalable from process development to production. Our comprehensive fluid management offerings allow you to be more confident with secure fittings and consumables. Repligen's goal is to drive integrated smart systems that can be PAT automated using our variable path-length technology. By integrating our inline FlowVPX systems into each system, our customers will have process insights like never before and be able to control their processes based on real-time analytics. The obvious issues with traditional UV methods include the assay time. This can take anywhere from 30 minutes to 3 hours depending on how many samples you're testing. This is certainly labor-intensive time as well, when we're talking about sample prep, dilution, standard curves, calculations. If you send samples internally to a different lab for testing and have to wait for those results, you can proceed with further processing. If you do in-process testing and have to send to QC for test results, then it's also the time that you have to spend waiting for those results. Sometimes there can be a long queue of samples QC has sent to test. Sometimes there's shifts in sample batches that you have to work around. But nevertheless, these are bottlenecks you have to deal with before you can get your results in order to make a decision to proceed with your next step in either PD or manufacturing. Manual dilutions and calculations certainly add more time and error to a validation process, thus delaying the implementation of the process altogether, adding more time and stress to the entire work. Dilution errors can increase significantly depending on whether you're diluting volumetrically or gravimetrically. And these errors can be further compounded and cumulative and can range between 5 to 20%. Traditional A280 protein quantification can take anywhere from 30 minutes to 3 hours, depending on the number of samples, number of dilutions, and pre-post calculation work required. Typically, the sample is taken from production line that needs to be transferred to a lab for the sample to be analyzed, then have that data sent back to production for them to react or to continue the process. It's not uncommon to hold production lines up to 8 hours whilst waiting for the results to come back from the lab. Dilution introduces errors. It's a manual process, takes a lot of analyst time, and requires dilution steps to be validated and regular testing of the analyst's pipetting abilities. Typical dilution error introduced, again mentioned, from the 5 to 20% error. The SoloVPE is the first and only at-line process analytics concentration measurement system that uses slope spectroscopy. To be very clear, this is still a UV-based technology, but it's an updated and monitored approach to protein measurements using our VPE technology. The results require no estimation, no dilutions, no manual calculations. Having a slope-based technique allows us to have more accuracy, better reproducibility, and the better data quality that allows us to have easier validations and a better implementation experience between inter and transfer methods. Variable path-length extension technology is also adaptable to inline analytics. Our Flow VPX system can be incorporated directly inline into your process for true inline monitoring, for true process analytics. So the real approach to slope spectroscopy is to refine the process and get us away from the 7-step process that is not only labor-intensive, but also has error-prone dilution steps involved. To really scale it down, we want to turn this method into a measure report function, which just takes a couple minutes. SoloVPE requires no sample dilution, or limited prior knowledge before testing, and no need for offline manual calculations of data. The SoloVPE generates a slope value for each measurement. This is built-in quality that can be assessed with every measurement. You generate your R-squared value for every single slope measurement required. Customers have shown that implementing SoloVPE into manufacturing rooms, clean rooms, can have significant advantages. SoloVPE is a simple method, a very simple system to run that can be easily implemented for production staff to perform directly in a clean room environment. The system is accepted by regulatory and GMP environments. It can be validated for your specific samples. We have over a decade of experience supporting all of our inline analytics within the GMP environment. The benefits for the ADC application and the capabilities of method creation will be to measure 2 wavelengths of interest without the need for multiple dilutions. So, what is variable path-length spectroscopy? Traditional UV-Vis-based spectroscopy based on Beer-Lambert law, where absorbance is equal to extinction coefficient times path length to equal your concentration. In traditional UV-Vis, the path length is fixed. You would particularly use a 1 centimeter cuvette because of your path length being fixed. The odds that you will have a limited concentration range available to you will require the performance of dilutions to be able to make that sample readable within the linear range of your spectrum. As mentioned before, Our technique is still UV-based. However, we are varying the light path, the path length used for our method. This means your sample can stay neat and resident within the sample vessel, and we will allow the system to automatically determine the most accurate path length to take and path length steps to generate linear data within the sample without user intervention. Fiber optic light brought into the SoloVPE is a solid fibrate. Light will be emitted through the bottom of the fibrate, so your path length is between the bottom of the fibrate and the bottom of your sample vessel. In both inline and at-line analytics, there's a stepper motor that moves this fiber up and down to vary the path length. As mentioned before, the SoloVPE is a UV-based technology. We use the Agilent Carry 60 as our light source, and using a fiber optic cable, we bring that light from the Carry 60 into the SoloVPE. Don't unvalidate any portion of the light source itself. We're purely using it as a way to bring the light from A to B. The order of operation is as follows. Once you've inputted your method information into the software, the fiber will lower itself to 0 position. This is enabling the fibrate to start at the bottom of the vessel. The next step will be the software algorithm scanning the wavelengths of interest from small path lengths to large path lengths, ranging anywhere from 5 microns up to 15 millimeters. The software is looking to detect one absorbance unit based on the absorbance change. Once the software finds the one absorbance range, it will then plot between 5 to 10 data points worth of regression data to provide to you the linear slope regression. Slope is the OD value that you'll be using for all your subsequent calculations after. Data acquisition takes less than 1 minute, and the evidence and validity of the sample will be based off an R-squared value of at least 3 nines or higher. That will become your pass-fail criteria, knowing that you have valid, accurate, linear data for the measurement. The software enables you to collect single, multiple wavelengths of interest and also offers full scanning capabilities using the variable path length technique represented by the spectra taken below. The wavelength ranges from 190 to 1100 nanometers, so the ability to apply slope regression data throughout the entire wavelength range is possible. This is really where the technology will shine for antibody-drug conjugates, where you're looking at the linker drug aligned with the therapeutic to understand what wavelengths of interest will we be targeting, and subsequently measuring both wavelengths of interest without the need for multiple sample dilutions in order to calculate concentration. This, applied with the current user results in the software, will also be able to allow the users to generate their DAR, drug-antibody ratio, results in real time without any sample manipulation to the neat sample. Additionally, the benefits of using slope spectroscopy do not limit us to the current issues that traditional UV or other fixed path length traditional UV options offer. In most cases, we are not baseline correcting or blanking for our samples. Simple reason being that all of our slope measurements are based on whether or not there is a physical change of absorbance over path length or not. Most formulation buffers do not exhibit absorbance change, meaning there's no reason to baseline correct or blank. Another added addition to the system, being slope-based, things like scratches on the vessels, fingerprints, don't impact the readings because there's no physical slope in your fingerprint, which makes this a very user-friendly technique when you're implementing in environments where potentially a trained scientist wouldn't be operating the system. Lastly, the majority of our business using variable pathlength technology is within the GMP environment. A few years back, Slope Spectroscopy and more importantly variable pathlength solutions were written into the ASTM standard for general practices of UV-Vis quantitative analysis. The statement reads, to avoid the dilution step, the instrument may contain an automatic system will allow the adjustment of path length measurement cell to optimize the measured absorbance. This is obviously meant to embrace the technology and show that a more accurate way to measure your protein samples, your antibody drug samples, a variety of different gene therapy modalities has a better way to show more accurate and repeatable results with removing all of the error-prone issues related to traditional UV. To summarize, our platform will increase process understanding and process development stages to accelerate the product to market through a variety of different groups. We are interested in reducing the cycle time and increasing our process efficiencies by eliminating delays associated with traditional offline testing. Repeatable, high quality, accurate linear results are the basis of our technology, with minimizing the risk of continued monitor throughout the process.
Thank you, Joe, for a fantastic presentation. I think our audience may have some questions, and that's okay. Like that, but I would like to encourage them to, I'd like to encourage the audience to submit their questions to us in the text box. And with that, I'd like to introduce for our second presentation, Priya Patel. Priya is a scientist at AbbVie focusing on high throughput and concise method evaluation and method development using screening technologies for biologics, including monoclonal antibodies, ADCs, and multispecifics. And throughout her career, first as a quality control chemist, then as an analytical scientist, Priya has gained valuable experience in detailed method validation, understanding of GXP regulation, and developing novel methods for challenging biologics. Priya, the floor is yours.
Thank you for the introduction. Hi everyone, my name is Priya Patel and I'm an analytical scientist at AbbVie working on method development of biologics, including ADCs. During my part of the webinar, I'll be going over some of the challenges we face when measuring protein content of ADCs and how we go about validating a solid slope spectroscopy method. At the end of the webinar, we'll have a live Q&A session where Joe and I can answer your questions. So don't forget to submit your questions in the chat. A main focus of AbbVie's evolving pipeline are antibody-drug conjugates, a class of potent biopharmaceutical drugs combining the targeting capabilities of monoclonal antibodies with the tumor-destroying properties of cytotoxic drugs. To evaluate the efficacy of the ADC, it's necessary to measure the protein concentration. But there are certain challenges associated with doing this, including but not limited to those listed on this slide. It's not as simple as measuring protein content for a monoclonal antibody because you can get interference from the drug or the linker at the same wavelength maxima as the protein. And this can be dependent on the drug load, so you need to carefully evaluate the extent of the overlap and apply a correction factor, adjusting this as the drug load changes. Another issue I'm sure we've all struggled with at some point is limited sample volume. Sometimes there's just not enough material to get all the necessary testing done, especially during early phases of development. It's precious material, so finding ways to save on that is highly beneficial. And lastly, as we move through the development process, the matrix the ADC is formulated in can change. Higher concentration formulations can provide patients with options of requiring fewer doses, but this can be a challenge analytically as it introduces issues with detector oversaturation and potential interference from new excipients. As Joe explained, a lot of these issues can be addressed using slope spectroscopy techniques. So slope spectroscopy methods are commonly used in our analytical labs, from early discovery through in-process manufacturing and down to our commercial quality control labs. With traditional fixed path length methods, analysts have to spend time diluting samples to an appropriate concentration, cleaning vessels to avoid cross-contamination, setting up instruments to run the calibration curves and baseline blanks. We'd need to use the same sample vessel for each measurement, so it made running batch samples almost a full-day process. With disposable vessels that are provided with the SoloVPE instrument, it's easier to move through those samples in much less time, since you can just fill the vessel and change it out with a new one for each measurement. This is so beneficial when performing in-process testing because that turnaround time can be cut quite significantly. It's also great that the same method can be used across a wide protein concentration range, so we can support conjugation from start to end without needing to adjust the method on the fly. And we don't just use this method for protein content measurement. We can also use it for drug-to-antibody ratio of the ADC by collecting absorbance data at the wavelength of both the protein and the drug linker and applying a dual wavelength correction. We can't, with this method, determine site specificity of the drug linker, but we can use this method to provide a quick overall DAR measurement. So how do we know our data from this instrument is reliable? Our methods go through a thorough validation process, including precision, accuracy, and linearity. For the data presented here, we used an ADC where both the monoclonal antibody and the drug linker absorb at 280 nanometers. The UV maximum of the drug linker is 370 nanometers, so absorbance readings were taken at both wavelengths, and a correction factor of 0.2 was applied to account for that absorbance of the drug linker at 280 nanometers. We targeted 3 different concentrations, the nominal with one above and one below. And to get to those target concentrations, we concentrated the sample using a spin column to 30.8 mg per ml, and then diluted down using the formulation buffer. We then analyzed each sample in triplicate using a new fibrate sample vessel and aliquot. So for this study, we set the acceptance criteria at 3%, and as you can see, this was met for each concentration. Something to note for the 120% sample is that the second measurement was slightly lower than the other two, which bumped that RSD up a bit. This was probably because the sample may not have been mixed correctly, so we'd recommend to mix by gently vortexing for about 10 seconds before taking a reading. I'd also recommend not letting the sample sit for too long before taking your reading to avoid any settling of that protein. The nominal sample at target 22.5 mg per ml had the best precision, but this is expected since that measurement was performed neat and there was no sample dilution or manipulation involved. The next study was accuracy, so we determined this using the same data set that we collected for precision. There's no need to collect additional reading. Recovery was calculated for each concentration level by dividing the measured concentration by the expected concentration and multiplying by 100. This study required the measured concentration to be within 95 to 105% of the expected concentration, which, as you can see, was met. Again, the neat sample at 100% concentration demonstrated the best accuracy as expected, and similar to the precision study, the 120% sample had the highest variability from the expected concentration because of that low second measurement. So mixing well is key. Linearity was the last study to be assessed, which we did across a range of 20 to 120%. As mentioned before, the target concentration samples were generated by first concentrating up and then diluting down in the formulation buffer. You might notice a difference in the measurement values for the samples at 120%, 100%, and 80%. That's because for this study, we measured the samples a couple of weeks later, so had to prepare fresh samples. But you can incorporate the linearity study into your precision study and use the average of the 3 measurements for each concentration, measuring additional samples for 50 and 20%. That way you can perform precision, accuracy, and linearity all in one experiment. The acceptance criteria for this study required the R-squared value to be equal to or above 0.995. Our observed R-squared value was 1.000, demonstrating great linear agreement with the expected concentration, and this really highlights the broad measurement range of the instrument. So the study plan we just went through is appropriate for stage 1 validation, but as you move to a commercial method, you'll need to assess additional parameters like intermediate precision, specificity, robustness, and range. With intermediate precision, sample handling can become quite important, as the way it's stored, mixed, and even the length of time the sample has been sitting out on the benchtop can affect your protein content measurement. So in conclusion, ADC protein content measurements using Slope Spectroscopy methods are simple to validate, with stage 1 validation only taking a couple of days, including review and verification time. They're so easy for the analyst to run compared to the more labor-intensive traditional UV-Vis approach, where there's more room for error, and can reduce the lag time in getting key results delivered. We're also exploring the feasibility of inline slope spectroscopy tools, so process chemists can get real-time data and make any necessary measurements quickly without having to wait on the lab analysts to deliver results. This comes with its own set of challenges, like finding an appropriate location to place the instrument and providing data equivalency to our current tools, but it's looking very hopeful.
Well, thank you, Joe and Priya, for 2 fantastic presentations. But I'm now sure that our audience has some questions, and with that, I would like to open up the opportunity for the audience. Actually, we already got some submissions of questions, and opening up the Q&A session.
And with that, we have a few questions. I would like to start with a question for Joe. Joe, is the SoloVPE system and software fully GMP compliant? And in addition, how are the system and software qualified, and how can CTech support the instrument, computer, and software qualification? Joe?
Sure. So yes, I think it's an important distinction that vendors make. Is their system compliant or achievable of compliance? 80% of our business in Repligen Analytics is GMP based. The software package that we've implemented for both versions we support offer the capabilities of full 21 Part 11 compliance. Our application specialists specialize in working with our users in post-sale support to help them implement. We also offer computer system validation as a service as well. So not only can you be assured that everything was done correctly, and we work with the analysts to set up the proper credentials and permission structures, but we can narrow that time frame down substantially by months by leveraging our experience to perform that validation versus training onboard IT to be responsible for the system and getting them trained on the software.
Right. And for Priya, if you look at the system, how long did it take you to install the device and to do the slope spectroscopy validation at AbbVie?
Yeah, thanks for the question. So getting the instrument up and running took less than a week, actually, for full installation and operational qualification. So once we had that instrument at our site, it didn't take long for our metrology team and Repligen field service engineer to work together to unpack it, get it on our network, get it qualified and calibrated, and test those connections out. So a very short amount of time.
Right, so that sounds very interesting. So, and then also for you, are there specific recommendations of best practices when implementing the technology? Following your presentation, I think you have something to share with us there.
Yeah, so there are a couple of additional recommendations, I guess, other than the mixing. So we keep a compressed air can on hand, and at the beginning of each set of experiments, we give the system a clean to remove any of that buildup of dust, and that could potentially interfere with the measurements and readings. Every month, we also perform the coupler check, and we remove the fibrette holder, clean it with mild detergent, and stick a fibrette through to make sure there aren't any obstructions, and make sure that mechanism is running smoothly. And we also power cycle every week just to reset the connections between our servers, the network, and the instrument, and make sure they're talking to each other as they should.
Okay. Now, a question that came in from our audience: is it possible to explain the possibility of DAR measurement in more detail?
Yeah, I can start this one. So, in this publication, we spell out the math and the algebra associated with it. What's nice about this analytical solution is, in our software, the calculations are built into the analytics. We specifically did this within the method, so the user does not need to go to any other external source like an Excel spreadsheet to make the final calculations for DAR. So this is completely built into the software. It's validatable, and the actual math, like I said, is listed within the software details and this publication.
Okay. And so another question that came in from our audience, is it possible to measure viscous samples with SoloVPE, and does it require special preparation?
Sure.
Joe?
So this is one of my favorite questions to be asked. The short answer is yes. It is certainly possible. It's actually pretty routine for us. The beauty of measuring a viscous sample is that you only need to make sure that the sample is pipetted, from your Falcon tube into one of the SoloVPE vessels. So as long as you actually just get the volume from A to B, that's all that's required. This obviously has a lot of benefits because you're not worrying about any residual residue left from potential dilution prep. So very easy, something typical. And again, a lot of our sort of experience with high concentration monoclonal antibodies where, once you start approaching 200 mgs and higher, samples can be quite viscous. Again, just another feature of our technology that you don't need to be concerned with.
Okay. And a question for Priya. For ADC concentration determination, how is the concentration factor determined? And in addition to that, is it something we need to know based on an expected DAR?
Yeah, good question. So what we do at AbbVie is we measure the concentration of the linker, the drug linker, across a range, and then we measure it at the absorbance wavelength of the protein and the absorbance wavelength of that drug linker, and then it's just a simple division, the absorbance wavelength of the protein divided by the linker. But DAR does have an effect, so you might want to get a sample across different DAR ranges and then perform that same calculation across those and measure those at different concentrations as well.
Okay. Now, Now, Joe, I know you explained something about the DAR measurement in more detail, but we have another additional question with that. Would you be able to provide a little bit more details on the value for an ADC can be obtained from this technology? Maybe a little bit add on to that.
Yeah, the only real add-on I could do is actually relay the algebra associated with the equation, which I certainly don't want to take the audience time to do that. I think one of the things we could potentially do is provide the link to the publication after this webinar that specifically has that information that you're looking for, or certainly you could reach out to myself or Priya after this, and we can give you, the physical content, of it.
Okay. So for our audience, you will receive an email about the presentation with additional information where you can meet what Joe said, obtain more information about this presentation and about the questions you may have asked. Okay, so another question that came in is what type of maintenance is required for the SoloVPE? I think a lot of people would like to know that.
Sure, I can take this one. Keep in mind this is still a UV-based technique. We have just obviously we're using the slope spectroscopy and the variable Pathlength as the preferred option here for the method. Typically, your weekly maintenance is down to about 5 minutes. We use, as I mentioned, the Carry 60 as a light source. So based on Agilent's recommendation of powering it down and turning it back on once a week. The other 2 tests would involve cleaning that delivery fiber that links the Carry and the Solo together, because that becomes the light source. That's simply done by unscrewing the connection and cleaning the surface of the fiber with a Kim wipe. And then you'd want to run what's called as a quick check transmission test, which just assures the proper amount of light is getting from the instrument to the Solo. And for bonus points, depending on what environment you're working within, you would run a system suitability standard on the Solo, just like you would for your typical processes, and you're off and running.
Right. So that seems to be a very simple approach to maintenance, right? Now, if you look at the SoloVPE software, does the SoloVPE software have a built-in functionality to support dual wavelength correction for concentration determination of an ADC?
Sure.
Yes.
I can. Go ahead.
Go ahead. I was going to say it does, and we actually use this quite a lot for our projects at AbbVie. We use it for quite a few of our ADCs. It's very simple to set up in the software.
Yeah, I'll just tack on to that. One of the great features within the software is that it is automated, right? As I mentioned, so there are 2 platforms of software. One that you program the math into the software, and then can save it as a method. That way, additional users or the next time anyone logs in, they can simply open the method, and none of this has to be done. You just run straight away, essentially load your sample and start. But we do have a specific software app for ADC concentrations where that best practice functionality, the equation for DAR, is hard-coded in the software. So all the user is required to do is put in the wavelengths of interest, extinction coefficients, and then press Start Collect.
Okay. So next question. What about a solution which easily have microbubbles and homogenizing it? What are your recommendations for an optimal measurements with a SoloVPE? And I think, Joe, you may be able to answer that.
Sure. SoloVPE samples are relatively low volume, right? So most samples for ADCs, the required volume, I'll play it safe to say anywhere from 20 microliters up to 100 microliters of sample. We certainly do care about the sample being homogeneous. What the Solo is very good at telling you is what it has at the time of measurement. So if your sample isn't mixed, the most typical result you see is a underestimation of concentration. What's really nice about microbubbles, we're typically dealing in microns of path length. There's virtually no chance for any of the microbubbles to interfere with the light path. Most bubbles that we've seen would be micro, they're at the top of the vessel anyway, where the light source being the fibrate is nowhere near to begin with. So that was the long answer. The short answer is not really an issue because of how our architecture of the measurement is set up.
Okay, thank you so much for that. Another question is, what is maximum concentration of ADCs that can be measured in a SoloVPE?
Well, we've tested concentration. Yeah, we've tested at least up to like 100 mg per mil or 120 mg per mil at AbbVie. You have to use the slightly larger vessels, but it's shown pretty good linearity up to those high concentrations.
And another question that came in, is it possible to measure oligonucleotide concentration in LNP by the SoloVPE? Maybe Joe, you can answer that question.
Yeah, sure. Great question. So yes, we have tested oligonucleotides before. We have a publication with Ionis Pharmaceuticals on that topic itself. Very, very concentrated. So obviously that's a great modality to potentially look at Repligen analytics for because of the abilities to go to very, very small path lengths. I will say regarding LNPs, stay tuned. We have applications and publications for oligonucleotides, plasmid purity, mRNA. We are in the middle of a project to work on the contribution from the LNP and sample to be able to do both in the same method. So the short answer is yes, you can do it. And within, I would say, the next 3 to 4 months, that evidence will be made public with the industry.
Okay. And I think we have one more question, and it is a little bit more about the equipment itself. Can the variable path length spectroscopy method be used to release of GMP finished drug products? And where do you see this biggest adoption of this technology? And that's a question also for Joe.
Sure. So as I mentioned previously, 80% of our business is in GMP environments. The technology is truly meant to be rolled out as a platform. We remove most of the error-prone elements, related to sample handling, dilutions, and limitations of traditional UV, where you don't necessarily need to be a trained scientist to operate the system. So certainly the biggest benefits for groups, has to be manufacturing, in QC, sort of downstream, where typically before we came along, you were waiting hours, by sending samples out for analysis and prep and waiting for those results. With both the inline and the at-line analytics that we offer, it's instantaneous results. So you're not waiting anymore, and certainly the validity of those results is proved via the R-squared value of the slope regression. So we've built in sort of an accuracy model to have evidence on just how accurate that data is.
Okay. And with that, I think this was our last question. Joe, Priya, do you guys have anything to add to our presentation today, or?
No, just thank you for listening in and for those good questions.
Okay. And Joe?
Yeah, thank you all for taking time out of your morning or afternoon to listen to us, highlight, this modality for slope spectroscopy. Certainly a special thanks, for Priya for not just the work that she's done, but certainly, working with AbbVie to make this evidence to the industry public. I think we can all agree that we certainly all feel a lot better when we've had sort of our collaborators in pharma take the baton and implement it themselves. So this is going to be the first of many for this particular modality, and continued, progression into automation will be coming. And certainly, if you have any questions, please download the article. Feel free to contact Priya and I anytime if you have any more specific questions we can help with.
Okay. And thank you, Joe and Priya. And I must say thank you for our audience to join us this morning. And again, on behalf of Priya and Joe, I'd like to thank you for doing so this morning. Now, as Joe mentioned, you may have additional questions. And of course, we like to answer these questions. So after today's webinars, you will receive an email with our contact information. And please feel free to also contact Joe and Priya and us to submit your questions via email. And again, we'd be happy to answer them. And again, thank you for joining us today. Please don't forget to tune in for our next webinar. Again, thank you all.